Okay, now that I successfully scared everybody away, I hope it wasn't too boring. I know it was more lecture-y, um, but it was one of those things where I was just trying to like really hammer in the key idea behind these things because I'm gonna be honest, I didn't hammer it in, so I'm trying to like have you guys learn from my mistake of not uh you know not really like holding myself accountable to make sure that I got those concepts 100% because when it came time to the exam, I fumbled it. I easily lost like 10 points from just the material I covered today. Like it was kind of bad. Um, but hey, we live and we learn. It wasn't the end of the world, but I definitely beat myself up because I was like, how am I ever gonna, you know, get an A in the class if I'm gonna just be like this? So I'm glad most people came back. Um, I remember when they were signing me up to be a pal, they were like, don't be discouraged if nobody shows up to your thing. So 4 people is good. I had 7 earlier, but I probably burnt them out. So it's okay. So any questions on the material that I kind of just covered? I know it's a little confusing to those who haven't been in class yet, but um, is there anything previously, like I can go all the way back to microscopes if you guys want me to. No question is a stupid question. Never. You want to go through the review questions? Yeah, we can. Totally. Okay, let me pull up my slideshow. And I somewhat went through this already, like just for the first couple ones. Man, why isn't this loading? Oh, here we go. Dang. Uh, so let me share my screen. I don't like the ads. So I sent out a couple of these on Remind. And this is, I think, one of the shorter ones. So we can breeze through this for the last little bit of the review session. And if you guys want to go through the other ones on your own, and if you find something that's like, I have no idea what this is, I can ask Dr. B if it's going to be a prominent idea on the exam or if you guys need to know it in detail. If you guys are ever too scared to go to Dr. B or if you guys want to find something out, um, that maybe you feel intimidated asking Dr. B about, I'm the middleman. I don't mind being the middleman. So let's just jump right into it. So we have our first question here. So which microorganisms are used to make microbiological growth media? What do you guys think the answer is for this one? Got a chat. A. Okay, I've got a couple A's. Everybody now. Got a C. Okay. C. Okay, so I think we're pretty much almost evenly split between A and C, at least with the answers I've gotten. So one party is correct and the other party unfortunately is not. So when we look at this question, I want us to ask ourselves, not what can we grow, but what is being used to grow. Okay. So for this question, it's asking us what is in a TSA plate, right? So um, if we were to look at the idea, what if a TSA plate was made out of bacteria? Well, remember that bacteria need nutrients to grow. And if there's only a certain number of nutrients, they can compete with each other and one will win, right? It's just like the animal kingdom. You know, if there's only a certain number of lions and only a little bit of food, only certain ones are going to survive. It's the survival of the fittest, right? So the one thing is that the agar is made with algae. Okay. Uh, if you say algae or algae, I don't mind. I just have, I just pronounce things weird sometimes, but C is correct for this one. Okay. So most commonly people will say A, but this question is specifically asking how do we make the plates? Not necessarily why do we use them, you know, the application that you might be more familiar with, because you guys haven't poured a plate before. So does that one make sense? Does it make sense as to why it might not be A? Okay. I got a yes. So I'm gonna roll. Okay. If you guys do have any questions about this, take a picture of it, send it to me and be like, can you explain this more? And I'll write it out if you need to. Okay, so next one we have Leeuwenhoek described microorganisms as, what did he describe microorganisms as? I've got a couple A's, a B. What are we thinking? I'm gonna double check this for myself because I don't want to be, I don't want to give you guys the wrong answer. Okay. Yes, the answer keys are on here. I've got a B. Okay. So this is more one of those things that's just like, um, you know how like you answer a question and it's like, you know, this is red. What is this? You know, it's one of those things that's like in the text, you kind of have to just look for it and know it. It's not really a theory type of thing. So there is going to be maybe a few questions in regards to some of the prominent people in microbiology, but animalcules is the correct one for here. So Leeuwenhoek described microorganisms as animalcules. Okay. There's not really much I can like, you know, like rationale with this. It's like he just said they were animalcules one day. I don't know why he did it. He just did it. Man, tough crowd. I hope you guys are laughing, but if not, that's okay. I promise I'll get funnier eventually. Alrighty. Thank you. Alrighty, so moving on to the next question. Which scientists first hypothesized that medical personnel can infect patients with pathogens? So we'll look at the people real quick. So Edward Jenner. So what did Edward Jenner do? You guys remember?
He introduced the aseptic technique, I think, or no?
I think you're thinking about Lister.
Lister, yeah. Jenner's the one that used the kid as like a
He did some
patient
shitty stuff to his maid's kid.
when trying to think of a vaccine.
Yes. So what he did was his landscaper's child. He took his landscaper's child and his milkmaid, So his milkmaid was infected with cowpox, and she had sores on her hand, and pus was oozing from said pores. He decided to inoculate the kid that's not his, it's his maid's, with said pus, and then demonstrated how he would be able to gain immunity from the pustules. So he basically demonstrated the idea of a vaccine, although our vaccines in this day and age are not to take pus from somebody and rub it into ourselves, but that's how he got the ball rolling, I guess. I wish it was different, but why would you do that to not even your own kid? It's like for science. It is. It's nefarious. I was like, why would you do that? Okay, so we have Edward Jenner. So we know that's not Edward Jenner. He did something totally crazy with some maid's kid. Um, Joseph Lister. Do we know what he did? You guys can look back in your notes. What did he do? I might pull up the.
He thought of the aseptic techniques and sterilizing his equipment.
So remember, key point is he sterilized his equipment, right? That was something that wasn't being practiced before, and he sterilized his equipment. What about Jon Snow? Do we know what he did? I could even just Google it.
He studied outbreaks and infection control.
There you go. So we have him. What about Ignaz Semmelweis?
He's the one who started the hand washing techniques and took the techniques from the midwives to prevent the pleural fever.
There you go. So that is so now knowing what we know. I know we've discussed what Jenner did, Lister, Snow, and Semmelweis. Who do you think first hypothesized that medical personnel can infect patients with pathogens?
Ignaz Semmelweis.
Okay, there you go. So the key thing is that with Lister, we would see that he sterilized his equipment, right? But when he sterilized his equipment, it wasn't necessarily personnel. You see where that differentiation is? Hand washing is that if I'm personnel and I touch a patient, I could give them whatever's on my hands. So that's personnel infecting patients. But if the question was, which scientist first hypothesized that medical equipment can infect patients with pathogens, then the answer would be Joseph Lister. And then another thing with Semmelweis, I think he actually got fired for his his obsessive need for everybody to be washing their hands. I think he got fired for that. Or he got his license revoked, something like that. So are we good on this one? Oh, shoot. But yeah, man. I would make like a Caitlyn or like Edward Jenner or whatever joke, but whatever. About them being evil or something. Okay. All right. So we'll move on to bacteria flagella. So bacteria flagella are, let's see what our options are. Anchored by the cell by a basal body. Okay. Composed of hemi. Okay. Surrounded by an extension of the cytoplasmic membrane. Okay. And composed of tubulin and hollow microtubules in a 9+2 arrangement. Okay. What do we think this is? If it's a shot in the dark, it's okay. We'll go over it. Let's see. I got an A. I've gotten an A. A's. Okay. Let me take a question versus 1, 2, 3, 4. Okay. Double checking. Okay. Yep, you guys are right. I couldn't think of it for life. So yeah, good job, guys. A is correct. So one thing that I think students could do is they could see C and say, oh, well, yeah, it's surrounded by an extension of the cytoplasmic membrane. But why is that not correct? Anybody know? So the reason why that's not correct is because that applies to eukaryotic flagella. Eukaryotic flagella is surrounded by an extension of the cytoplasmic membrane, but bacterial flagella is not. Okay, so you guys saw past it or whatever, but you got the right answer. That's what counts. But good job, guys. Alrighty, so moving on to the next one. If you guys are good. You guys okay? I'm gonna take that as a yes. Why is Spotify trying to open? Okay. Yes? Okay, yes. So next question is, which of the following is the most associated with diffusion? Okay. This one can also trip up people. What do we think? This one has a lot of, I think. Just kidding. You guys are on it. Everybody's saying C and everybody's correct. So good job, guys. So one thing, the one thing that gets that students get tripped up with is endocytosis. They think diffusion into the membrane, but they forget about out of the membrane too. But also endocytosis, it can be it could be something that is like just kind of flying by the membrane. So like if it's a lipid soluble item, if it's something small, it can go through the membrane itself. But carrier proteins is what is most associated with diffusion. The key word is definitely most in this case. But yeah, good job, guys. Let's move on. So endospores, this question about endospores. So collect all that information about endospores in your brain. So endospores are reproductive structures of some bacteria, occur in some archaea, can cause shock, fever, and inflammation, or are dormant and resistant cells. What do we think? Man, I couldn't even finish saying it out, and people are hopping on it, but you guys are right. You guys are right. It's D. Yep. So I think of these like endospores are like the scariest things to me because they are like, I don't know, it's like the trump card. They're like, you think you can kill me? Psych, here's my trump card. And they'll just create endospores and survive anyways. Like, you're kidding. How is that possible? But yeah, so endospores are dormant resistant cells. Good job. Alrighty. So now we have gram-positive bacteria. So gram-positive bacteria, let's see, have thick cell wall, which retains crystal violet dye. Okay. Contain teichoic acids in their cell walls. Okay. Appear purple after gram staining, or all of the above. See, I have a D. Okay. I think I got mostly D's. I'm gonna double check something because the wording of this has me like thrown off for some reason. Let's see, it's gonna be number 7. Yeah. Okay, yeah, you guys are right. The thing that was, I guess, like a learning moment, is the specific wording of cell wall. So some, like for me, cell wall and like the membrane, they trip me up all the time. So you guys saw past that. I'm glad. Um, but that's what tripped me up. Because I was like, is this correct? Because you know, they have a thick peptidoglycan. I know that, but cell wall, I don't know. So good job. You guys all saw past it. They all came to the same consensus. Good, good, good. All right. Which cellular structure is, oh, and by the way, I'm sorry, we already started into overtime. Before I do the next question, if you guys do need to leave at any time, you can. I'll go through, man, already. Okay. I'm sorry. But I'll go through a few more of these if you'd like. I'm available for a while, but if you guys need to go, you guys can go. And I'll just kind of talk until there's nobody here. And in like probably 20 minutes, if you guys want to stay and go that long, I'll do another break. So I'm already getting some. So which cellular structure is important in classifying a bacterial species as gram-positive, gram-negative? That's pretty straightforward. So you guys said B, which is correct. I'm gonna double check that for you guys because sometimes my confidence wavers. So yes, you guys are correct. There we go. So yeah, I think we all pretty much have that drilled into our brains. So moving on. So resolution is best described as the, okay. So remember that really tedious long lecture about resolution and other things like that? What do we think? I have a B. Okay. I have a C. Discourse. I think I have, yeah, I have a B and a C. So let's look at what we can do. I want to make sure that I didn't tell you guys the wrong thing. Okay. So the answer is going to be C. So remember, resolution is kind of how clear we see something, right? So if I were to give you a ping pong ball and a golf ball, they're both white balls, right? And if I put them up, against maybe a surface that has more contrast, right? A surface that has more contrast than this, from afar, you wouldn't have very good resolution. You'd just be like, those are two white balls. But if you get closer and you have better resolution, you'd be able to distinguish, oh, okay, this is a smooth ping pong ball and a coarse or rough golf ball. That resolution allows you to make that difference. Allows you to see that difference. So when you magnify something, technically, I suppose that with greater magnification comes greater resolution. But again, this is one of those questions that asks, how is it best described? Although you're not completely wrong, you're in the right field, you're just not where the question wants you to be, if that makes sense. So you guys are on the right track. Just try to, slow down. It's not always like, how fast can you, do it? So like, one other thing I did want to mention about test taking strategies for this class, one thing that really helped me was that I wrote everything out. So if it asked me about gram positive or gram negative, I drew out the two cell walls next to each other. That way I wasn't gonna get confused. I wasn't gonna be like, is that, I can't remember if that's gram positive, gram negative. I would just think, what does a gram positive look like? Gram positive, draw it out. What does a gram negative look like? Gram negative, draw it out. That way, if somebody asks me a question about that, I can look at it and I can visualize it and I won't get scrambled in the membrane. If that makes sense. I wanted to mention that before, because we are technically like kind of doing a simulated, a little more relaxed, but exam type of thing. But yes, so does everybody understand what resolution is? And maybe you guys can look back on that. One other thing that I did want to show you guys, this practical study guide right here. Follow it, live by it, devote your life to it. If you do this and you understand all of the concepts in lab, you will do well on the lab portion of the exam. Okay? This is my wink wink. Look at this and take advantage of this. It helps, and like when you look at these things, don't just be like, oh yeah, I know what a gram stain is, move on. Even if you know what a gram stain is, you write out what happens, what you do, and you also write out the differences in the two and understand how each step works. So that's my wink wink to you guys. All right. And I think there will be a couple things like, oh, define resolution, define this. So you guys will be fine. Okay, in the binomial system of nomenclature, which term is always written in lowercase letters? This is really niche. Let's see, D. I'm gonna double check it. I agree with you guys. Yeah, it's D. You guys are good. I feel like you guys understand lecture more than you do lab, where I'm the other way around. Like somebody, like, I don't know how you guys do it. Read one thing and understand immediately. I don't know. It's crazy to me. Okay, I think we're all good on that one. It's another one of those things. It's like it's in the text somewhere. So I'm gonna move on to redox reactions, right? So redox reactions. Remember these from chemistry? I have a C already. And then I have a D. Okay, D. If you want to, I'm pretty sure I know what this one is. Woo. I double check it because you can never be too sure. Yep. Man, I'm jumping all over the place. Sorry, I don't mean to make you guys seasick. So if you answered C, technically correct, but there's an all of the above. And I know those suck so bad. It's an ultimatum, and sometimes you just don't want to have to deal with ultimatums. I get you. It's one of those questions, but you are right. But slowing down and taking a second to, look at them. I promise you the 3 hours that you're gonna get for an exam is enough. You'll be okay. Sometimes I would go like, sometimes people only took an hour. I don't know how they're built like that, but I couldn't just take an hour. I felt like I would miss something. So you can take the full 3 hours. Most of the time, nobody went the full 3 hours. So you take advantage of it. You're supposed to be there for those 3 hours. You can be there the whole 3 hours. She's not going to kick you out. But if you want to ever like go over your answers like a second time when you're taking the exam, do it. My only thing is that if you're like torn between two answers and you circled one the first time, do not erase it. Just stick with it. If you have a 50-50 chance, stick with the 50-50 chance you initially thought. Okay. All right. Let's move on. So coenzymes. Oh, I hope I can get through this. So coenzymes are, what do we think these guys are? We have type of apoenzymes, proteins, inorganic cofactors, or organic cofactors. So I believe I've gotten 2 Ds, 3 Ds, and 2 As. 3 Ds and 2 As. Okay, 3 Ds and 2 As. I believe it's D. Yep, it is D. I think this is another one of those things that it's somewhere in the exam one content. I'm pretty sure it's going to be either in here or it's going to be in anatomy, because this metabolism one is like strictly like fermentation and stuff like that. So yeah. Also, I'm not too familiar with this, so that kind of speaks to how big of a topic it was, at least for my exams. That could be different for yours. But if you guys don't understand something going on, I'm more than willing to brush up on it. Let's see. So does anybody have any questions that I could answer here? I want to ask you guys, why do you say apoenzymes? Was it like a shot in the dark, like that was the biggest word you saw and you're like, yeah, let's go with that? Or did you guys think differently? Because I know that's sometimes how I think. I'm like, that's a big sciencey word, and I don't know that other big sciencey word it was talking about, so I'll just put those two sciencey words together and hope. And no shame in that. Kind of want to find it now. I mean, you probably did. I mean, it's entirely possible. I'm gonna run it open real quick and try to look at it. Although it'll never let me just try to find it quickly, but I'll try my best. Hope I got another chat. Oh yeah, no problem. Head out whenever you guys need to. I think at 6:20, I'm probably gonna cut it off, and I'll finish up the last few questions on this one. Probably just on Wednesday. Let's see. Yeah, I can't quite find it, but man, that kind of sucks. Is there any other questions that I could answer in the meantime while I'm trying to find this answer for you guys? Or if you guys just know it.
Yeah. I just wanted to ask a question because I remember in one of my like my at-home specimen, like the one that I took home. So what I realized is I understand like the shape that it looks like, but its arrangement, I'm literally not sure like what it's like. Not arrangement, but maybe it's not like strepto. You know what I mean? It's also not like staph because it's bacillus. So I just was wondering like maybe if you could look at it and like I don't know, like help me like.
So it really just depends on kind of what, because obviously your gram stand is gonna reveal what it looks like. But if you were to take a picture at like of your bacillus bacteria or oxide bacteria right in the middle where it's like the densest, it's gonna look like staph. And it's always gonna look clustered if you go to the thickest parts. What you try to do in terms of like identifying something in a gram stain, you want to go towards the edges and you want to try to look for the like if you were able to see something like a single arrangement by itself in a clearing, that's what you want. Like that's how you're going to try to identify it. So what I would say is if you can't see distinguishable chains, I would say that they're most likely going to be single bacillus. because it's just if you guys already did the gram stain and if your culture's already thrown out, because I think we did that last week, we just don't have enough room in the fridges to keep everybody's cultures all the time. But if it's, I would say just single, or
Okay,
you can try to say Diplo and like scoot by with it.
okay. That's
but yeah.
helpful, honestly.
Okay,
Yeah.
I'm glad.
Okay. Thank you.
Yeah, no problem. Yeah, I wanted to mention to you guys, on Friday from 1 to 4, there's going to be a basically makeup lab where you can practice your streak for isolation, you can practice your gram stains, you can ask Dr. B questions, you can sit there and just review and study if you'd like. All of those things are going to be available to you, and I will be there. So if you need to ask me questions, you can. So yeah, whatever you guys would like to do, I'll be there for you. I'll be there for you. Like the Friends theme song. Ha ha ha. But yeah. So if you guys would like to go, it's not required, but you can definitely go if you'd like. But yeah. It's open for you guys. And you guys, Monday, Wednesday, and Tuesday, Thursday, both welcome. Like you guys don't have to fight over a spot or anything. Let's see. Okay, I think I found it, by the way, the answer to our problems for this one. So I'll explain this really quickly and then I'll just call it quits because I know everybody's got to probably go somewhere and dinner's right around the corner. Why is it so small? What the heck? That's okay, there it is. Okay, so we were talking about how what our coenzyme is, right? So it's a non-protein portion and it's an activator, right? So coenzymes are going to be organic, okay? So these are organic. Cofactors are inorganic. So that's why we might get confused between the two. And apoenzymes is the protein portion. Okay, so we saw the apoenzyme. It combines with the coenzyme and a substrate in order to produce a reaction. And our coenzyme can be organic. Okay. Our coenzyme is organic, but it can also be replaced with a cofactor, which is inorganic. So they both play the same role. It's just one's organic and one's inorganic. Okay. So I'm going to stop it there. I hope that we had a great review session. And I will see you guys hopefully on Wednesday. Okay, guys? My Zoom's about to kick me out. So bye, guys.
Bye,
Thank
good
you
night,
for coming.
Lauren.
Bye.